So, good afternoon to all. I am Dr. Mahesh from belonging to INSTEM and NCBS. And today I am talking about the CRISPR genome editing, generating the mouse model for the human therapeutics. All your, my voice is reaching to all? Last, yeah, sure. So, madam already told how the designer babies and all. So, we are generating the designer mice for the various application of studying the human therapeutics and all, and that's we are routinely generating the 10 to 12 mice models every year, and that's we will learn here today how we are generating the mice models, right? Yeah. So. We are getting around 300 crores of the base pairs, nucleotide base pair from our parents. Half is from father, half is from the mothers. And these are the genetic building material that is passed on from the genetics to genetics. So these are called like a chromosome, genes, and the proteins, all you know, the central dogma and all you are studying in the biotechnology, right? So these our genome is constantly under the threat of the DNA damage, and every day a lot of DNA damage are happening in the our body, and that is called the various in the form of the double standard break or the various breaks, and the these DNA damage can be repaired by our own body itself. We have the cellular machinery, and that is repairing that. That's the cell and making it functional. But those DNA damages are not repaired, will be accumulated, and that turns in the form of the impaired cell function, and that is result into diseases. So, for example, earlier also people, the speaker has told about the sickle cell anemia. So, it is a single base pair mutation. So, these one base pair mutation are causing the entire blood cells into the in the form of the sickle shape, and impairing the functions of the body. And there is a no cure for such type of the diseases until now, after discovering the so many drugs and all these things. So to treat such type of diseases, researchers are in search of the new technology, and that's how the programmable nuclease come into picture. Right. So recently, three programmable nucleases are come in the picture. The one is the ZAPN, another is TALEN, and the last one is the CRISPR. And due to its simplicity and the affordability, CRISPR is making the boom, and there is a lot of good products are coming to treat the diseases with the CRISPR-Cas system. So what is a CRISPR? CRISPR is the bacterial immune defense mechanism system that is adapted as a gene editing tool. So when the bacteria is attacked by the viruses, the bacteria will procure some of the SNPs from the DNA, from that viruses, and incorporate into their genome. And the next time the same virus is attacking on that bacteria, he will use as a reference sequence of that genome what is incorporated earlier, and it will create an immune system. So bacteria saving himself from the attack. So these tools has been developed as a this tool has been adopted from the bacterial system to use as a genetic editing tool, right? So only the Cas9 protein that we are not synthesized by the human body system has been procured from the bacteria, and with the help of that, the human genome or the any genome, including plant and the any other species, can be manipulated, right? So this is like a 25-year of the research that the different scientists has done over the period of time, and the one element has been discovered, and finally the all pieces has been gathered in 2012. The professor Jennifer Donda and the Emmett Carpenter has shown that with the help of these guide RNA and the CRISPR-Cas system, we can create a double standard break into the human genome. And that is what the great discovery they have shown. And that whatever the double standard break is happening in the genome can be repaired by our system. So that's how we can correct the gene. Right. So in the last 10 years, the lot of, 12 years, lot of research has been done, the lot of gene knockout and the plant gene edited crops or the different type of the base editors or the CRISPR editing system has been come. And the future is like that. It will be currently the sickle cell anemia FDA has approved the sickle cell anemia therapy using the CRISPR, and with the help of the imaging or the artificial intelligence and the sequencing, the things are advancing a lot, and we are getting the cure for the diseases. Right. So currently, just I told now the sickle cell anemia, there is already a FDA has approved the CRISPR therapy in the UK and US, and the other various diseases are under pipeline. So what is the use of these therapies? So earlier managing the sickle cell anemia, patient has to take a lot of medicine and lot of pain, but by using these type of the therapies, we can, even if we go, the patient can walk to the clinic and within a one shot of injection, he can get a permanent cure. So that's how the quality of the patient's life as well as their near and dear ones' life or the will be well-being, welfare will be happen. So in our lab, we are trying to develop the models, and the mice is used as models for studying the various genetic diseases, and it's the basic laboratory tools. So we are using the CRISPR-Cas technology and we are generating the various model. So this is the CRISPR is very simple and the affordable and the turnover is rapid. Hence, from 2012, the model generation using this technology has been increased a lot. And this has, we have adopted at our place and we are generating the model for any type of these things. So what we need to create a double stranded break at the genome, we need a guide RNA called 20 base pair recognition site that will search into the genome and find the target. And that's we need also the PAM sequence. These are the details maybe you can find and go by Google. So by using this thing, there are two types of the mechanism. One is the non-homology enjoining and another one is the HDR mediated direct repair mechanism. So by using both by these two different mechanisms, we are generating two different type of the models. Like for knockout, we have to use this non-homology end joining mechanism, as well as for knock-in model or the point mutation, we have to use the homology directed repair mechanism model. And this is the workflow. With the help of the bioinformatics, we are designing the strategy, which gene at what location and what point we have to make the changes, and then the reagent will be synthesized in-house or procured from the companies and then micro-injected into the one-cell embryos or two-cell embryos and generate the models and screen the models and screen the pups, whatever we are getting, and establish the lines for that. So how this work, some scientists will find out, the researcher will find out some mutation in the patient that is novel mutation, and that's they want to correlate or study into the human, they validate into the model. So they are giving us that particular things, which type of the mutation they want to create into mouse model, and we will take over the project. So how difficult it is to generate a mice model? So it's very simple. With the help of some bioinformatics knowledge and the molecular biology tools, we can easily able to generate this thing. So first thing is we have to find out the target. Here I have given the example of the knockout animals where I want to knock out this exon 2 or like a particular critical exon that is contributing for the protein expression or what? First, yeah. So with the help of that thing, we can just generate the design, and by using the free resources, we can find out the guide RNA that is essential for this type of the studies. And then we can synthesize the reagents, validate the reagents into the laboratory, and this is our 4-day protocol where we are injecting the embryos and making the gene editing possible. Right, sure. So here is how we are injecting with the help of the micromanipulator. We are injecting the embryos with the particular cocktail of the solution, and the DNA edit will happen. And likewise, we are generated a various model, including some point mutation, and that are showing the phenotype every year. So due to lack of time, I have to go pretty much faster. So in the COVID, we have generated the model called ACE2 model, and the various laboratory has used this model for validating their vaccines, as well as the some COVID-related molecules also. And we are also doing the assisted reproductive technology at our place, where we are cryopreserving the models, and we have the whole sorts of more than 700 mice model that different researcher can be used at different places. So we are combining these two facilities, animal research facility and the mouse genome engineering facility, where we are organizing all these things, and we have the very high barrier facility for animal model. And that's how we are conducting the workshop and seminars also. Those who are interested, they can join for such type of workshop and get trained with us. Yeah. Thank you very much.